Welcome to the Virtual Scholars Day presentation of Hayden Bowman. Just a little housekeeping before we get started. Please make sure that your mic and video are off, but the sound on your device is on so that you are able to hear me and our presenter today, Hayden Bowman. Hayden Bowman is presenting her honors thesis as part of her requirements for the Carl Goodson Honors Program. Hayden is a biomedical science major. This presentation and others can be found at OBU Scholarly Commons. I will now turn things over to Hayden, who can tell you a little bit about herself before presenting. Hayden, are you ready to share your screen?
Yes, I'm good. Can you hear me?
Yes.
Perfect. Yes, I'm ready. Okay. Maybe there. Okay. Well, before we get started, a little bit about me. Like Miss Autumn said, I'm a biomedical science major. I'm from Searcy, Arkansas. And following this year, graduation, whenever that may be, I'll be attending medical school at UAMS in Little Rock. Okay, am I good to begin?
You sure are. It looks great.
Perfect. All right, so welcome to my honors thesis presentation. This is over a research project that I did last summer at the Arkansas Children's Research Institute, which is the research wing of Arkansas Children's Hospital. I worked with Dr. Graham Strube in the ENT department, and our project is over the identification and characterization of microRNA in the pathogenesis of lymphatic malformations. So what exactly is a lymphatic malformation? Well, these are congenital anomalies that are most prevalently found in the head and neck area. They're consisting of ectatic lymphatic channels. So basically your lymph channels that are just growing and there's no check on where they grow, how they grow. It's just uncontrolled. These lesions can be macrocystic, microcystic, or mixed in nature. And they're really significant because they have the potential to be life-threatening due to the compression infiltration of underlying critical structures. So for instance, take the picture on the screen. These lymphatic malformations are in the neck area, so they could be compressing the main arteries and veins that bring blood to the brain or your airways. So studies have shown that there are potential mutations within a gene called the PIK3CA gene that are a potential driving factor behind this aberrant lymphatic endothelial cell proliferation and the lymphangiogenesis that is seen in this disease type. So here's an image that shows the different types of lesions that can occur. So microcystic is just a bunch of small different pockets that are combined in the same general area, and it looks large, but there are a bunch of different individual cysts basically. And then macrocystic is just one large lesion. So for this study, we specifically wanted to look at the use of molecular therapy as a treatment. And the reason that we wanted to do this is that these lymphatic malformations are very difficult to treat surgically due to their location and like how they grow in and around the different structures. There are inadequate medication options. So there are some that are available to help treat and kind of dial back the amount of growth that's present, but nothing actually prevents it or stops it fully. And then even if they are able to be removed surgically, there's a very high recurrence rate. So they're very likely to come back afterwards. In looking at different types of molecular therapies, we specifically wanted to focus on microRNA. And so this is just a short video about microRNA expression. So microRNA begins just as any other type of genetic material inside the nucleus of every cell. Your DNA is opened and then a ribosome will attach, transcribe it into an RNA molecule. This molecule will then be folded into a hairpin precursor. And once it's in this form, it's actually able to exit the nucleus into the cytoplasm of the cell where it can take its microRNA function. So a splicing occurs and a 15 to 20 base pair segment of single-stranded RNA is formed, and that's the microRNA. MicroRNA has the ability to function, so it will actually bind complementary to a strand of mRNA. So the mRNA is the copy that your proteins are produced from. And when mRNA binds to it, it actually prevents the protein from being made. In using this, we actually want to use this almost against it. So we would like to treat this mRNA with a microRNA inhibitor. And so what this would do, as seen on the right, is it would bind to that microRNA and prevent it from binding to the mRNA molecule. So if you leave it untreated, that microRNA will bind, prevent protein expression. But when you treat it with an inhibitor, it allows that ribosome to actually make the protein. Now, this plays into what we want to look at because some of the microRNAs have the ability to act as a tumor suppressor silencer. So basically, genes in your body that act to prevent cancer and a lot of growth rapidly, these genes would prevent that. But in some things, microRNA can inhibit those and actually cause the growth to continue unchecked. So for our study, we wanted to evaluate whether lymphatic malformations expressed abnormal microRNAs comparatively to the normal tissue, and whether this caused the growth that is seen through an activation of a downstream signaling cascade. So in order to do so, we collected tissue samples from 4 different patients. We ran a background experiment to confirm the expression of known micro and mRNAs. Then we profiled the expression of microRNAs known to participate in the pathway that we were targeting, and we determined the effect of any abnormal microRNA findings on the downstream pathway components. So the first thing that we did in our experiment is we performed a background RT-PCR screening of lymphatic malformation tissue and normal skin samples from multiple different patients. And so in doing this, we screened for 4 known mRNAs and 2 known microRNAs that are found in lymphatic endothelial cells. And the purpose of this was just to establish a validation that the experiments and the procedures that we would be doing in our study were valid and that we could actually find what we were hoping to find. So after we had validation that what we were wanting to do would work, we moved into examining the PIK3CA gene and its downstream pathways. So we ran a Western blot, which is an analysis of protein expression. And so what we found when we looked at this downstream PIK3CA pathway is that of the multiple different protein targets that we looked at, only MAPK was phosphorylated at a higher percentage in lymphatic malformations compared to the normal tissue samples for all 4 patients. Some others were differentially expressed, but we weren't quite sure what that was for. But the fact that we saw a consistent dysregulation among one of these downstream proteins between all 4 samples, and it was consistent between the 4 samples, it allowed us to realize that yes, there is a potential mutation within this pathway. And then we wanted to look at what exactly that would be. So from there, we looked into the different microRNA that are actually known to regulate the PIK3CA pathway. And from a literature review, what we found is we found 6 different microRNAs, which are listed on the left side of the screen. And we screened those. We did RT-PCR for all of these on the samples that we had. Now, what we found is that from the 6 different microRNAs that we screened, only microRNA 21 had a consistent upregulation in the lymphatic malformation compared to the normal skin. Now, there were no other patterns that were able to be determined from this. So we decided that we wanted to further explore microRNA 21 and the implications that it has on this regulation of this pathway. MicroRNA 21 happens to be one of the most studied oncogenic microRNAs in literature. Numerous studies have been published showing that it targets tumor suppressor genes that are associated with proliferation, apoptosis, and invasion that are associated in this PIK3CA pathway. Now, the image on the left shows just how many ways that an increase in microRNA 21 can have an effect on its downstream targets within this pathway. So it acts on the tumor suppressor genes and allows those to be knocked out, which promotes that growth that is seen. The picture on the right is a study that we hope to recreate. So this study looked at microRNA 21's involvement in a different pathway and was actually able to show that using inhibitors and mimics, it was able to control that tumor growth as seen by the tumors shown. Our current progress, we're doing ongoing experiments. They're still doing experimentation with this at Children's, looking at multiple different microRNA 21 regulated targets in these paired samples that we have. And a few of them are listed below, most of which are tumor suppressor genes or proliferation factors. In the future, this study will hopefully transition into in vitro models using lymphatic endothelial cells and including either the presence of microRNA 21 mimics or inhibitors in order to determine the effect of microRNA 21 on cell growth. They would also like to use cell sorting to isolate differential cell types in order to confirm just which cell types microRNA 21 production actually occurs in. That way it will allow for better targeting. And finally, they would like to develop a method of in vivo delivery of microRNA 21 inhibitors as either a primary or adjuvant therapy to help treat these lymphatic malformations. Now, at the end of the summer, I was very intrigued by molecular therapies and their use in medicine and the potential of developing these strategies out to help different disease types as medicine progresses and as the medical world comes into more technology. How are these exactly applicable? And so in order to do that, I decided that I wanted to do an individual study on these molecular therapies. And it just happened that Alzheimer's disease and type 2 diabetes kind of played into my hands. So my grandfather is a type 2 diabetic, and he shortly after the summer began showing signs of Alzheimer's disease. Now, I actually remembered sitting in on different lectures that discussed Alzheimer's disease having a biofilm. Now, a biofilm is a type of organic structure, basically like a spider's web almost, but it occurs in your body with different types of proteins where they aggregate and basically become a film. One thing that they mentioned was that you need an abundance of sugar to promote this formation. And so I began to wonder if there was any correlation between diabetes, Alzheimer's disease, and this formation of a biofilm which potentially drives Alzheimer's disease. So during the course of this study, I learned that Alzheimer's disease has been reported by the World Health Organization to consist up to almost 70% of all different types of dementia. And Alzheimer's disease is driven by two main different proteins, the beta amyloid protein and tau protein. Beta amyloid functions normally under normal circumstances to aid in neuroplasticity, synapse formation, memory formation, and homeostasis. Unfortunately, under abnormal circumstances, it cleaves into a longer peptide than normal, which makes it basically get stuck to itself, in essence forming what is called senile plaques, which is basically a type of biofilm. And also the tau protein. Tau is a microtubule stabilizing protein. So normally it is known to regulate neurite outgrowth and axonal transport within the brain. However, under abnormal phosphorylation, this promotes the formation of neurofibrillary tangles, which also contribute to that biofilm type formation. Now, between these two proteins, there's actually a very unique interaction that occurs. It's been studied, but there's no definitive proof that it happens one way or another. There's still a lot to be learned. But in 3 studies that I found, study number 1 showed that an increase in amyloid beta caused an increase in tau, but this was a one-way reaction. So if you increase tau, there was no definitive increase in the amyloid beta. A second study showed that amyloid beta contributes, it promotes a tau pathway, which leads to synapse dysfunction. So if there's amyloid beta present, it basically causes tau to make your synapses, which is the linkage between neurons in your brain. It causes them to not be as effective as they are initially. Also, a loss of tau exhibits no pathological function on the brain. A third study showed that if you remove the tau genes, you prevent any tau formation at all, that it actually becomes a type of neuroprotection. So the loss of tau actually benefits your brain and its health over time. This study also showed that if you have an overabundance of amyloid beta combined with an increase in tau protein, it leads to an increase in the senile plaque formation and actually promotes that biofilm type formation within your brain, which causes the progression of Alzheimer's disease. Now, type 2 diabetes, most people will know about this, but or know someone with it. This is a disease of the pancreatic beta cells. It's the most common form of diabetes mellitus and occurs when the body develops an insulin resistance. So your body still produces insulin, but it's unable to recognize that insulin, therefore it doesn't take up the glucose within your body as well. So the current treatment in most cases is dosages of insulin in order to stimulate the uptake of glucose through specific glucose translocator proteins. Now, for this study, we looked at two different glucose translocator proteins, GLUT1, which is found in every cell within the body, and GLUT3, which is located specifically in the brain. So in order to tie these together, we looked at the comorbidity of Alzheimer's disease and type 2 diabetes, focusing mainly on insulin resistance. So insulin resistance is the major symptom of type 2 diabetes, and it has the potential to lead to increases in many different Alzheimer's dementia progressive pathways, as seen in the chart below. So IR is insulin resistance, found on the lower left side. So if you have insulin resistance, then that leads to a few different progressions. It promotes a higher glucose concentration in the blood, the cerebrospinal fluid, and cells don't take up this glucose as well, so they lack the glucose they need to produce energy. This abundance of free glucose actually stimulates the biofilm formation. So insulin resistance also leads to low NMDA receptor activation. NMDA is similar to glutamate, which is a signaling hormone that your body uses within your brain. And this leads to high free calcium and high free glutamate. So the high free calcium is found in senile plaques during most autopsies. There's actually found to be a higher concentration of calcium within those areas. And this high calcium also has the potential to lead to long-term depotentiation, which means that over time, if there's high calcium, your neurons are less likely to interact and form synapse formations, therefore leading to the dementia type effects. Now, this high glutamate actually becomes neurotoxic. So if you have a high concentration of glutamate within your brain, then it's actually bad for your neurons and it causes them to basically die away. A third cause or pathway for insulin resistance is you're left with high levels of free circulating insulin. So in the blood, cerebrospinal fluid, there's lots of insulin that's not able to be used because your body isn't recognizing it. Now, insulin and amyloid beta are degraded by the same degradation enzyme. So when you have lots of insulin, it competes for the same enzyme that amyloid beta needs to be broken down. Now, recall that amyloid beta is found in the formation of those biofilms and the senile plaques that are a main contributor to Alzheimer's disease. Now, this competition for the enzyme actually produces an overproduction of amyloid beta, which further compounds the effects of Alzheimer's disease. So, being that there are no cure and very few treatments available for Alzheimer's disease, two of which that were studied recently are cholinesterase inhibitors and NMDA receptor inhibitors in methyl D-aspartate. These are more downstream than insulin resistance. So insulin resistance basically promotes the pathways that those treatments are targeting. So what we wanted to do is take it a step further and try to target the insulin resistance to get ahead of all the downstream proponents of that pathway. So we looked specifically to target GLUT3 mediated uptake of glucose as a treatment for type 2 related Alzheimer's disease. And so the proposed study that we had at the end was a two-pronged strategy. Number one, we wanted to develop an in vitro modification designed to increase glucose uptake through the GLUT3 translocator protein. And then the second step was to identify the best manner in which to target protein expression in order to elicit goal one. So there are multiple different ways to increase a protein's function, but we need to figure out what exactly is the best one for our specific strategy. I would like to acknowledge a few people that helped me along the way real quick. For the research I did this past summer on microRNA, this is my mentor and research team at Arkansas Children's Research Institute. And then for the Alzheimer's and diabetes research, I would like to thank Dr. Ruth Plymell for being the director of my study and for helping me along the way, answering any questions I had to the best of her ability, and kind of giving me guidance as to what to do next. All right, thank you. Any questions?
I don't think we have any questions right now, Hayden. Thank you so much for such an informative look at some possible ways to help individuals with these illnesses and problems. Thank you very much.
Yeah, thank you.
And thank our viewers for watching this virtual Scholars Day presentation.
Yes, thank you very much for coming to hear my thesis presentation. I hope that you found it informative, and feel free to ask any questions that you have. I'll try to get back to you.